And we suddenly realized, of course, in a joint lab meeting with Giselle that she had this wealth of microarray data from the various bone marrow aspirates that you heard from Pam. And we went and we asked Jeff Bond, and Julie Dragon did the analysis going back through Giselle's microarray data. And what we compared here is the relative level of expression on a probe that encodes chRNA5 for normal, this would be the normal sympathetic ganglion. These are cells isolated and propagated in cell culture from bone marrow aspirates that were not tumor initiating. And there are two different types of tumor initiating cells that can be isolated. There's a TIC S2 population and a TIC S1 population. And here are those FACS-sorted cells that Pam just told you about. And all three of these tumor-initiating cell types, actually these two tumor-initiating cell types are elevated with respect to chRNA5 expression, and they're comparable to the FACS-sorted cells. And in fact, if you actually sit down and examine the p-values and compare, for example, the non-tumor initiating cells with one subpopulation of tumor-initiating cells, you can see there's a 6-fold upregulation. The S2 population is the one that, in terms of gene profiling, most closely resembles the FACS-sorted cells that Pam told you about. And here you can see that the S2 subtype is 19-fold upregulated in comparison to the non-tumor initiating cells. Again, we're always talking here about chRNA5. And as well, the FACS-sorted cells show 7-fold upregulation. These are very, very highly significant p-values. In addition, here is the data from another subunit of a nicotinic receptor. That's the CHRNB2 that I showed to you. And this one shows an even more interesting profile. Again, here's normal sympathetic ganglion, the non-tumor initiating cells. Here's a subtype of the tumor initiating cells that actually doesn't have a gene expression profile that looks like the FACS-sorted cells. And they're also low in this subunit, but both of these populations, which seem to be more representative of true neuroblastoma cells, are highly upregulated in this particular gene as well. And here is the fold differences in, again, the non-tumor initiating cells with respect to these different subpopulations. And here, now you can see this S2 population. There's a 3-fold upregulation in CHRNB2 with respect to the non-tumor initiating cells, and as well a 3-fold upregulation in the FACS-sorted cells versus the non-tumor initiating cells. And again, extremely highly significant p-values associated with these differences. And then finally, we thought we should really look as well in primary tumors. And we did a pilot study with COG where we were able to obtain 20 different RNAs from primary neuroblastoma tumors. And on this, what we show here, again, this is relative expression. The normal sympathetic ganglion over here where the arrow is, that's 1. So that's normal. And everything is expressed relative to the normal sympathetic ganglion. And you can see, with the exception of maybe a couple of samples down here, every single primary tumor is showing hugely upregulated expression of CHRN5. This is 400-fold higher than a normal sympathetic ganglion. This is a subunit that's not very highly expressed normally in a sympathetic ganglion. And you can see here we've got very high levels, many hundreds of fold higher. So what is the significance of this gene being so upregulated in tumors as well as in tumor-initiating cells? I'm very interested in what might be going on here. Of course, this might not mean anything at all. It might be an epiphenomenon. It might be just something that goes up and is correlated with the fact that it's in a neuroblastoma cell. I prefer not to think that because, of course, I want to keep moving forward and asking what is really going on. Is it possible that this highly upregulated subunit is the target of MG624? And what is possibly the role of these nicotinic receptors in tumorigenesis? Now, what's really a puzzle here is that the alpha-5 subunit by itself is not a ligand-binding subunit. It's a weird alpha. All the other alphas can bind acetylcholine and participate in forming a functional ligand-gated ion channel in one of those pentamers. Alpha-5 can't do that. So if you take alpha-5 and beta-2 together, they can't act like a nicotinic receptor. So what could be going on? Is it possible that such high levels of alpha-5 can prevent normal nicotinic receptors from forming, and maybe the normal ones are the ones that are allowing in too much calcium and promoting proliferation? Is it possible that it could participate in forming a novel nicotinic acetylcholine receptor? Maybe it pairs with a different alpha, for example, alpha-7, and with beta-2 to form a functional subunit. Or does this gene by itself have a novel function that has nothing to do with normal nicotinic receptor function? So we're working on this model, and of course I'm very open to excluding this model, but here's what we think might be going on. Based on the panel of gene expression we see in the tumor-initiating cells, we see alpha-5, we see alpha-7, and we see beta-2. So this is a very rare subunit. This actually has been observed in the brain. It's not in the periphery. Sympathetic neurons normally have alpha-3 beta-4 or alpha-3 alpha-5 beta-4. Perhaps this is something that is hit by MG624. Now, MG624 is not a good drug to use on kids. I would not recommend it because it is a pan-nicotinic receptor receptor antagonist. It will royally mess up their autonomic nervous systems, but there's a possibility of modifying this so that you find something that is a lot more selective. And if this is a really rare subunit, functional unit, that is not normally there in the peripheral nervous system, and you can keep this drug out of the brain because if you make it hydrophilic, it won't cross the blood-brain barrier, maybe this is a novel new therapeutic that could be developed for targeting tumor-initiating cells in neuroblastoma. I'm very interested in pursuing this project, and I'm very excited to report, all of this work was done out of my own pocket. I've been funding my own research because it's hard these days to find money to do research, but I did submit NIH R21 on this project, and on Friday I got my score. It's the 10th percentile. I'm very excited about it. We have to make sure that Congress does not cut the NIH budget anymore because it's funded by NINDS. Their cutoff right now is the 14th percentile. Many other institutes have already dropped below the 10th percentile. So I would not be fundable in some other institutes of NIH. The NINDS staff tells me that unless Congress does do a drastic cut of the NIH budget, that they will hold at the 14th percentile next year, but we don't know because all of these negotiations are happening right now about the cap on the deficit. And that cap in the deficit is supposedly being negotiated for more severe cuts in spending. So this is a big political fight right now. But right now, I'm very hopeful that we can continue on this. What we want to do, the R21 is really simple. Take RNAi and knock out alpha-5 and see if we lose the sensitivity to MG624, see if we lose that transformed phenotype. Do a gain of function. Force expression of alpha-5 in these normal crest precursor cells and see if you get a gain of function, see if you turn them into a neuroblastoma type cell. So hopefully, I can't move forward until this money comes in, but hopefully I'll be able to do that. So with that, I would really like to thank my collaborators in this project. Giselle Scholler, who started this whole thing off. Up until Giselle came in, I got my head buried in chicken embryos, and I was happy looking at molecules that guide development of neurons. David Kaplan, who's been very generous with his tumor-initiating cell lines. He's a co-investigator on this project. If it gets funded, he will be providing the lentiviral constructs for the RNA targeting that we're going to be doing. Jeff Bond, who together with Julie Dragon helped sort of reanalyze specifically for the chRNA5 the microarray data that Giselle had. Cecilia Gatti, who is going to be a co-investigator on this R21, she's the one who's got all the great antibodies against alpha-5, antibodies against these nicotinic receptors that are highly selective, are very hard to come by. She's going to help us figure out exactly what kind of pentamer is expressed there. And John DeWitt is an MD PhD student in my laboratory who did all of these analyses for RNA expression levels in the various cell types I just told you about. So with that, I'd be happy to answer questions.
Thank you.